Welcome back to the deep dive. Today, we are taking a really close look at something fundamental to how our bodies fight disease.
We are.
We're going beyond just general defenses. We're talking about the body's own internal security system, the way it checks ID, basically, how it tells us from them.
Exactly. We're diving into the Major Histocompatibility Complex, or MHC.
MHC.
Right. And honestly, if you want to understand anything deep about immunity, transplants, allergies, autoimmune diseases, you have to get your head around MHC.
It sounds pretty central. You called it a badge reader, a security scanner.
Yeah, because that's essentially its job for the T cells, the main soldiers of our adaptive immune system. MHC dictates pretty much everything those T cells are allowed to see and react to.
Okay, adaptive immunity, that's the specific, powerful response system, the one with memory, mediated by T cells and B cells.
That's the one. Develops more slowly, but packs a real punch.
But you're saying the T cells, these specialist soldiers, they're a bit kicky. They don't just attack anything.
They're incredibly picky and kind of blind in a way. A T cell can't just, bump into a virus particle floating in your blood and attack it.
Really?
No, it needs that viral particle, or rather, a little piece of it, to be formally presented to it, held up on a special molecular platform for inspection.
Okay, so the T cell needs a formal introduction to the enemy, basically.
You could put it that way, yes.
Yeah.
And that platform, that molecular pedestal, that's the MHC molecule.
Got it. MHC is the display stand, but the name. Major Histocompatibility Complex. It sounds, well, very technical, maybe a bit old-fashioned.
It does, doesn't it?
Oh.
And the name actually tells you where it came from. It wasn't discovered because scientists were looking for the immune system's display stand.
Oh.
No, they found these molecules because they're the product of genes that cause the body to violently reject transplanted organs.
Ah, histocompatibility. Tissue matching.
Exactly. Histo for tissue, compatibility for matching. The immune system just goes haywire if it sees tissue with different MHC molecules. In humans, we often use the term HLA, Human Leukocyte Antigens.
HLA. Why the different name?
Same system, just a more clinical name.
Yeah.
They're first identified on leukocytes, on white blood cells, so MHC, HL, same critical function, showing peptide fragments to
No.
T cells.
Okay. MHC or HLA, it's the display. But you mentioned genetic identity, and this is where it gets really complex, doesn't it? The HLA haplotype.
Oh, absolutely. This is one of the most variable parts of the entire human genome. The genes coding for these HLA molecules are all packed together on chromosome 6.
Right.
But the key thing here is the extreme polymorphism. We're not talking a few variations, we're talking thousands of different versions alleles of these MHC genes across the human population.
Thousands. Wow. So the chance of two random people having the same set.
Is incredibly small.
Yeah.
Vanishingly small, unless you're identical twins. You get one whole set of these genes, a haplotype from your mom, and another complete set from your dad.
So my MHC profile is basically unique.
Pretty much, yes. And this massive diversity is, well, a double-edged sword. It makes our species resilient, but it's also the reason why finding a matched organ donor is so incredibly difficult. Your body sees those different MHCs on the donor organ as a major red flag.
That really sets the stage. It explains the name and the transplant issue. But now the mechanism. How does this display system actually work day to day? You said it needs different strategies.
It does, because threats can come from different places. Is the danger inside the cell, like a virus multiplying or maybe the cell turning cancerous? Or is it something the cell picked up from outside, like bacteria?
Two different scenarios.
Two different scenarios needing two different display systems. And that's what we have. Class 1 and Class 2 MHC molecules. Two parallel surveillance systems, each tailored for a specific job.
Okay, let's break those down. Class 1 and Class 2, where should we start?
Let's start with Class I MHC. Think of this as the internal threat monitor.
Internal monitor.
Yeah.
Checking what's happening inside the cell itself.
Exactly. Class I's job is to display peptides, little protein fragments that come from proteins made within the cell cytoplasm.
So it's like sampling the cell's own manufacturing line.
Perfect analogy. It's constantly showing snippets of what's being produced. Is it normal self stuff? Or is it maybe a piece of a virus protein or a weird protein from a tumor cell?
Okay. And if it's monitoring internal health, where would you expect to find these Class I molecules?
Everywhere. Well, almost everywhere. The source material really emphasizes this. They're on all nucleated cells and platelets.
All nucleated cells. So basically every cell except red blood cells.
Pretty much, yeah. And platelets, too.
Yeah.
Which makes total sense, right? Any cell with a nucleus could potentially get infected by a virus or become cancerous.
So every cell needs the ability to wave a flag if something's wrong inside.
Precisely. It's a system for self-reporting distress.
Okay, let's get into the structure of this class I molecule. You said it's a heterodimer, two different parts.
Yes. It's made of two protein chains. There's a big one called the alpha chain or heavy chain. This one is highly variable polymorphic between people.
That's where the genetic difference comes in.
Right. And then there's a smaller second chain called beta-2 microglobulin. This one is crucial, but it's not polymorphic. It's basically the same in everyone.
Why is that smaller one needed if it's not involved in the variation?
It's essential for the whole thing to work. The big alpha chain on its own isn't stable. It needs the beta-2 microglobulin to kind of prop it up, help it fold correctly.
Like a chaperone protein.
It's exactly like a chaperone. It binds non-covalently, and the complex of the alpha chain and beta-2 microglobulin is what can actually hold a peptide and get transported to the cell surface.
Yeah.
Without beta-2 meter, the class I molecule basically doesn't make it.
And the alpha chain, the variable part, which specific HLA genes make that?
For class I, there are three main genes: HLA-A, HLA-B, and HLA-C. They're all highly polymorphic, meaning lots of different versions exist in the population.
So I inherit one HLA-A, one B, and one C from my mom, and one of each from my dad. 6 class I alleles total.
That's right, giving you potentially 6 different types of class I molecules on your cells.
Okay, structure down. Now, the most important part, the peptide binding site, where the actual piece of protein is displayed. What's that like on class I?
It's formed by parts of the alpha chain, specifically the alpha dollar and alpha 22 domains. They fold together to create a groove, or cleft.
A groove to hold the peptide.
Yes. And, critically, for class I, this groove has closed ends. Imagine a little pocket.
Closed ends. What does that mean for the peptides it can hold?
It means it can only hold relatively shortened peptides, typically around 8 to 11 amino acids long. The peptide has to fit snugly inside that closed groove.
Okay, so it's quite specific about the size.
Very specific. And the amino acids lining that groove, those are the polymorphic ones, the bits that vary because of your specific HLA-A, B, and C genes. They determine the exact shape and chemical properties of the groove, and therefore which specific 8 to 11 amino acid peptides can bind.
So my HLA type dictates exactly which internal protein fragments my cells can show off.
Precisely. Now, how do those fragments get there? The processing pathway.
Right. Let's say a cell gets infected with a virus. It starts making viral proteins inside.
Okay, so those viral proteins, along with all the normal cellular proteins in the cytoplasm, are constantly being sort of recycled or disposed of. They get fed into these large protein-degrading machines called proteasomes?
Proteose, like cellular paper shredders.
That's a great way to think of it. They chop up proteins into small peptide fragments.
Okay, proteins shredded into peptides in the cytoplasm. But the MHC class I molecule is being made elsewhere, isn't it? In the endoplasmic reticulum.
Yes, exactly. The class I alpha chain and the beta-2 microglobulin are synthesized on ribosomes and enter the ER. So the peptides need to get from the cytoplasm into the ER.
How do they cross that membrane?
There's a dedicated transporter complex called TAT transporter associated with antigen processing. It specifically pumps these short peptides, generated by the proteasome, from the cytoplasm into the ER lumen.
Okay, TT delivers the peptides to the ER. What happens then?
Inside the ER, the newly made class I alpha chain is waiting, kind of held by other chaperone proteins. It binds to beta-2 microglobulin, and then it waits for a peptide delivered by TEAP that fits its specific groove.
The peptide binding stabilizes the whole complex.
It's the crucial final step. Once the right peptide binds, the whole MHC class I peptide complex becomes stable. The chaperones let go.
And then?
Then this stable loaded complex gets transported out of the ER through the Golgi apparatus and finally arrives at the cell surface, displaying its peptide cargo to the outside world.
Ready for inspection. Now, who does the inspecting? Which T cell is looking for this class I signal?
This is where the specificity is critical. Class I MHC complexes are recognized exclusively by CD8 plus T cells.
CD8 plus T cells, those are the cytotoxic T lymphocytes, right? The killer cells.
That's them, the CTLs. Their job is to kill cells that are displaying the wrong peptide signs of infection or cancer.
And how is this restriction enforced? Why only CD8 plus cells?
It's structural. The class I alpha chain has another domain, the alpha 3,3 domain, which is non-polymorphic, same in everyone. This domain has a specific binding site for the CD8 coreceptor molecule found on the surface of CTLs.
So the CD8 molecule on the T cell acts like a key fitting into a lock on the class I molecule.
Exactly. If a T cell doesn't have CD8, it can't effectively dock with the class I molecule, even if its main T cell receptor recognizes the peptide. The CD8-MHC interaction provides essential stability and signaling.
That's elegant. The variable part holds the message. The constant part ensures only the right recipient gets it.
It is. It ensures the killer cells are directed specifically to cells signaling internal trouble.
But wait, if class I is on almost every cell and they're constantly showing peptides from normal self-proteins, why don't our CD8 plus T cells just kill all our healthy cells?
The million-dollar question. That comes down to self-tolerance. It's absolutely crucial.
How does that work?
During T cell development in the thymus, there's a rigorous selection process. Any baby T cells whose receptors bind too strongly to self-peptides presented on self-MHC molecules are normally eliminated or inactivated. It's called negative selection.
So the T cells that graduate are essentially trained to ignore self presented on class I.
Broadly speaking, yes. They tolerate normal self-peptides. They only get fully activated and turn into killers when they encounter a class I molecule presenting a foreign peptide, like from a virus, or sometimes an altered self-peptide, like from a tumor, and usually need additional danger signals, too.
Okay, so tolerance prevents mass destruction, and the outcome when a CTL does recognize a foreign peptide on class 1.
The CTL gets activated, binds tightly to the target cell displaying the problematic peptide, and kills it. Directly eliminates the source of the internal threat, the infected cell or the tumor cell.
Simple and brutal. Effective. All right, that's class 1, internal surveillance, present on almost all cells, talks to CD8 plus killer T cells. Now let's switch gears completely to class 2 MHC molecules. You call them the external intelligence reporter.
That's right. If class I is about what the cell itself is making, class 2 is about what the cell has encountered and captured from the outside environment.
So presenting evidence from extracellular stuff like bacteria floating around or toxins.
Exactly. Things that have been internalized by the cell through processes like phagocytosis or endocytosis. Class 2 displays fragments of proteins that originated outside the cell.
Okay, different source material. Does that mean they're found on different cells compared to class 1?
Yes, absolutely. Their distribution is much more restricted. You find class 2 molecules mainly on what we call professional antigen-presenting cells, or APCs.
Professional APCs. Who are they?
The main ones are macrophages, B lymphocytes, and especially dendritic cells, DCs. These are the immune cells whose job it is to patrol tissues, gobble up foreign material, and then travel to lymph nodes to present that material to T cells.
So only the dedicated intelligence gatherers get to use the class 2 system.
Pretty much. It makes sense you want the cells whose job is raising the alarm about external threats to be the ones presenting that evidence.
Okay, let's look at the structure of class 2. How does it compare to class 1? Still a heterodimer?
Still a heterodimer, yes, but with important differences. Class 2 consists of an alpha chain and a beta chain. But here's the key. Both the alpha and beta chains are polymorphic.
Both are variable, unlike class 1 where only the heavy chain was.
Correct. Remember class, I had the variable alpha chain and the constant beta-2 microglobulin? Class 2 has a variable alpha chain and a variable beta chain contributing to the structure and the peptide binding.
And which HLA genes make these class 2 chains?
These are encoded in a different part of the MHC region on chromosome 6, called the HLA-D region. This region has several subregions like HLA-DP, HLA-DQ, and HLA-DR. Each subregion codes for alpha and beta chains.
So again, inheriting different DP, DQ, and DR alleles from each parent creates huge variability in the class 2 molecules a person can make.
Exactly. Massive diversity potential here too.
How about the peptide binding groove on class 2? Is it like the class I groove?
Structurally, it's also formed by domains from both chains. The alpha-DAWID and beta-BALAD1 domains interact, but there's a crucial difference. The class 2 peptide binding groove has open ends.
Open ends? Class I had closed ends. What does that mean?
It means class 2 isn't as strict about peptide length. It can accommodate longer peptides, typically ranging from 10 up to 30 amino acids, or even more. The ends of the peptide can hang out of the groove.
More like a channel or a trough than a pocket.
That's a good way to picture it, yes. Like a hot dog bun holding a longer sausage. This ability to bind longer, more varied fragments is suited to sampling the diverse proteins found outside cells.
Okay, structure clear. Now, the processing pathway. If class I started with cytoplasmic proteins and proteasomes, where does class 2 start?
It starts with the APC engulfing something from outside, a bacterium, maybe some foreign protein. This material gets taken into the cell inside vesicles, endosomes, or phagosomes.
So captured in a bubble inside the cell.
Right. These vesicles then mature and become more acidic, and they fuse with lysosomes, which are full of digestive enzymes. These enzymes chop up the ingested proteins into peptide fragments.
Okay, so peptide generation happens inside these endosomal vesicles, not in the cytoplasm like for class 1.
That separation is absolutely key. It ensures that peptides from internal proteins end up on class 1 and peptides from external proteins end up on class 2.
But wait, the class 2 molecules themselves are proteins. They must be made in the ER, like class I molecules. How do they avoid picking up those cytoplasmic peptides that TAP is pumping into the ER for class 1?
Excellent question. Nature has a clever trick for this. When class 2 alpha and beta chains are first made and assembled in the ER, they immediately bind to another protein called the invariant chain, or 2.
Invariant chain, what does that do?
Its main job is to physically block the peptide binding groove of the class 2 molecule while it's still inside the ER.
Like a placeholder or a cap.
Exactly. It prevents any peptides floating around in the ER, the ones meant for class 1, from binding to the class 2 groove.
Clever. So the class 2 molecule travels out of the ER with its groove protected.
Yes. The class 2 invariant chain complex moves through the Golgi and is then targeted to the endosomal compartments, the same place where the external antigens are being chopped up.
Ah, so it meets the peptides in that specific location.
Correct. Once inside those vesicles, enzymes start to chew up the invariant chain itself. It gets degraded, leaving just a small fragment still sitting in the groove called CLIP, class 2 associated invariant chain peptide.
CLIP, still blocking the groove.
Temporarily. Then another specialized molecule helps to remove CLIP and allows the peptides generated from the external proteins within that vesicle to bind to the now vacant class 2 groove.
Okay, so it's a carefully managed swap invariant chain off, CLIP off, external peptide on.
That's the process. Once a suitable peptide is loaded, the class 2 peptide complex becomes stable and is transported to the surface of the APC.
Ready for inspection again, but this time who's looking at the class 2 display?
Class 2 MHC complexes are recognized exclusively by CD4 plus T cells.
CD4 plus T cells. These are the helper T cells, aren't they?
That's right. The orchestrators, the coordinators of the immune response, not the direct killers.
And is there a similar core-receptor interaction enforcing this, like CD8 for class 1?
Yes, there is. The CD4 co-receptor on the helper T cell binds specifically to a non-polymorphic part of the class 2 molecule, specifically the beta-2,2 domain.
So again, the structure dictates the interaction. Is CD4 binds class 2, CD8 binds class 1, ensures the right T cell talks to the right MHC?
Absolutely. It guarantees that helper T cells are activated by APCs presenting external intelligence.
And what happens when a CD4 plus helper T cell recognizes its specific peptide on a class 2 molecule on an APC? What's the outcome? It doesn't kill the APC, right?
No, its job isn't to kill the messenger. Its job is to get activated itself, and then help other immune cells do their jobs better.
Help how?
Activated helper T cells can, for instance, provide signals that tell B cells to start producing large amounts of high-affinity antibodies against that external pathogen. Or they can release signals that superactivate macrophages, making them much better at killing the microbes they've ingested.
So class 2 presentation leads to coordination and amplification of the response against external threats.
Exactly. It's about tailoring the response.
Right.
Internal threat, seen on class 1, kill the cell directly, CD8. External threat in class 2, activate broader defenses like antibodies and empowered macrophages, CD4.
This segregation principle, as the source calls it, really is ingenious. 2 pathways, 2 sets of T cells, 2 different outcomes, all dictated by where the antigen came from and which MHC molecule displays it.
It's the foundation of adaptive immunity, specificity, and power.
Okay, we've got the mechanics down. Class 1, Class 2, CD8, CD4. Now let's zoom out. What are the bigger picture consequences of this incredibly complex and variable MHC system? You mentioned the genetics earlier, that massive polymorphism.
Right. Why on earth would evolution favor such incredible diversity in this one genetic region? It seems overly complicated.
The source suggested it's about survival of the species, didn't it?
That's the leading theory, yeah.
Yeah.
Often linked to something called the Red Queen hypothesis, the idea that we're in a constant evolutionary arms race with pathogens.
Meaning microbes are always changing to evade us.
Exactly. They evolve fast. If we all had the exact same set of MHC molecules, a single new virus or bacterium could potentially evolve a way to make proteins whose peptides none of our MHC molecules could bind and present effectively.
And if the T cells can't see it,
The pathogen could sweep through the population unchecked.
devastating.
But with thousands of different MHC alleles scattered across humanity,
The odds are much better that at least some individuals in the population will have an MHC variant capable of presenting peptides from that new pathogen. Even if 90% of us are susceptible, maybe 10% have the right HLA type to fight it off effectively. So polymorphism protects the species by ensuring some individuals can always mount a response. It's like hedging our evolutionary bets.
Precisely. It's population-level defense through genetic diversity, insurance against pandemic wipeout.
That makes sense. But this genetic lottery, as you called it, must have consequences for us as individuals, right? This links to disease susceptibility.
Directly. This is the clinical reality. Your ability to fight off a particular infection or even your risk of developing certain other diseases is tied to the specific MHC alleles you inherited.
How so?
Well, think about it. You could only mount a T-cell response against an antigen if your MHC molecules, the specific HLA-A, B, C, DP, DQ, DR variants you have, can physically bind and present a peptide fragment from that antigen.
So if I encounter a new bacterium, but none of my MHC molecules are a good fit for any of its peptides.
Your T cells might never get properly alerted. You could be much more susceptible to that specific infection compared to someone whose MHC profile can display its peptides effectively.
Wow.
Okay.
But does it work the other way too? Can having MHCs that bind certain things be bad?
Yes. And this is where it gets really interesting with allergies and autoimmune diseases.
How does that work?
Let's say you inherit a class 2 variant, maybe a particular HLA-DR type, that happens to be really good at binding and presenting a peptide from something totally harmless, like ragweed pollen.
Okay.
Your helper T cells see this pollen peptide presented very efficiently by your APCs, and they mount a strong response. But it's an inappropriate response against something harmless. That's the basis of allergic hypersensitivity. Your genetics make you prone to overreacting.
So my immune system's efficiency in presentation can actually cause problems if the target is wrong.
Exactly. And this becomes even more critical in autoimmunity. The initial surge of interest in HLA genotyping clinically came from discovering strong links between inheriting particular HLA alleles and the risk of developing autoimmune diseases.
Diseases where the immune system attacks the body's own tissues.
Right. The thinking is that certain HLA variants might be unusually good at presenting self-peptides that should normally be ignored. Or perhaps they present foreign peptides that look very similar to self-peptides, leading to cross-reactivity. The failure of self-tolerance seems strongly influenced by the MHC background.
Are there well-known examples of these HLA disease associations?
Oh, absolutely. Some are incredibly strong. The classic one is HLA-B27.
B27, that's a class I allele, right? HLA-B locus.
Correct. Inheriting HLA-B27 dramatically increases your risk for a group of inflammatory diseases called spondyloarthropathies, especially ankylosing spondylitis, a condition affecting the spine.
How strong is the association?
It's huge. Something like 90% of people with ankylosing spondylitis carry the HLA-B27 allele, while it's only present in about 8% of the general population.
That's a massive difference. What's the theory there? Why does B27 predispose to this?
It's still debated, but a leading idea is molecular mimicry. Perhaps the B27 molecule presents a peptide from a gut bacterium, maybe, that looks structurally very similar to a protein found in our own joints or spine.
So the T cells get activated against the bacteria, but then mistakenly attack the self-tissue because it looks the same.
That's one prominent hypothesis, yes.
Yeah.
The unique structure of the B27 binding groove might facilitate this cross-reaction.
Any strong examples involving class 2?
Yes, definitely. Certain HLA-DR alleles, particularly variants of HLA-DR4, are strongly linked to an increased risk for rheumatoid arthritis and also for type 1 diabetes.
Both autoimmune diseases. And DR is class 2, talking to helper T cells.
Exactly, which fits because these diseases are thought to be driven significantly by inappropriate helper T cell activation against self-antigens. The specific DR4 variant might be particularly adept at presenting the relevant cell peptides in joints or the pancreas.
It really underscores how our personal MHC code shapes our health landscape, for better or worse.
It absolutely does.
Okay, which brings us full circle back to the original discovery transplantation, the MHC system as the major barrier.
The absolute major barrier.
Yeah.
It all comes down to that HLA haplotype we talked about, the complete set of HLA-A, B, C, DP, DQ, DR genes you inherit as a block from each parent.
And because there are so many variations, the chance of two unrelated people matching is tiny.
Extremely tiny. Even between siblings, the odds aren't great. Remember, you get one haplotype from mom, one from dad. Your sibling gets one from mom, one from dad. But they might get the other one you didn't get from each parent.
Right, leading to the famous statistic.
The 1 in 4 chance that two siblings will inherit the exact same two haplotypes, making them a perfect HLA match. There's a 1 in 4 chance they share no haplotypes, a complete mismatch, and a 1 in 2 chance that they share one haplotype, a half match.
Which is why siblings are always the first potential donors screened for things like kidney or bone marrow transplants.
Absolutely, because the closer the HLA match, the lower the risk of the recipient's immune system attacking the donated organ or cells, what we call graft rejection.
What exactly is the recipient's immune system reacting against in a mismatch? Is it peptides or the MHC molecules themselves?
It's primarily the foreign MHC molecules themselves. Even if they're presenting perfectly normal self-peptides from the donor tissue, the recipient's T cells, both CD4 plus and CD8 plus, recognize the structure of the foreign MHC molecule as non-self.
So the T cells don't even need to see a foreign peptide. The foreign display stand itself is the target.
Yes, this is called allele activity. T cells that were trained on your own MHC molecules can directly recognize and react strongly against different MHC structures. And because MHC molecules are on almost every cell, class 1, or densely expressed on APCs infiltrating the graft, class 2, the immune attack is powerful and widespread.
Leading to graft rejection.
Exactly, which is why we need HLA typing before transplants and why recipients usually need lifelong immunosuppressant drugs to dampen this low-reactive response, even with a decent match. It just highlights how central MHC is to defining immunological self.
Okay, this has been an incredibly detailed journey through the MHC. Let's try to boil it down, synthesize the absolute key takeaways for everyone listening.
Right. At its core, MHC molecules are the body's essential display system for T cells. No MHC presentation, no T cell response, period.
They're the mandatory ID checks.
Pretty much. And there are two main systems, working in parallel but handling different types of threats.
First, class I.
Class I MHC, made by the HLA-A, B, and C genes, found on nearly all your nucleated cells. Its job is internal surveillance. It shows short peptides from proteins made inside the cell.
And it reports, too.
It reports to the CD8 plus cytotoxic T cells, the killers. The message is essentially, here's what I'm making inside. If this looks foreign or cancerous, kill me.
Okay, and the second system?
Class 2 MHC, made by HLA-DP, DQ, and DR genes, found mainly on professional antigen-presenting cells, like dendritic cells, macrophages, B cells. Its job is external intelligence. It shows longer peptides from stuff the cell ingested from outside.
Reporting to?
It reports to the CD4 plus helper T cells, the coordinators. The message is more like, look what I found out here. Activate the right defenses, get the B cells making antibodies, get the macrophages powered up.
So the big takeaway is this brilliant division of labor, isn't it? The MHC system ensures the immune response is tailored, kill infected cells directly for internal threats, coordinate broader defenses for external threats.
Exactly. It's all about using this molecular language, the MHC peptide complex, to direct the right type of adaptive immune response to the right place at the right time. Specificity and coordination.
And it all hinges on the specific MHA genes we happen to inherit.
That genetic lottery, yes.
Which brings us to a final thought, maybe something a bit provocative. We've seen how our specific HLA type influences our fight against infections, our risk of allergies, our susceptibility to devastating autoimmune diseases like ankylosing spondylitis or rheumatoid arthritis, linked to alleles like B27 and DR4.
Right, those connections are powerful.
So, given that MHC is so central to presenting specific peptides, both foreign and self, what kind of future medical possibilities open up if we could learn to somehow tune or manipulate this personal MHC display system?
That's really the holy grail for a lot of immunotherapy research, isn't it? Imagine if you could selectively block your specific autoimmune-linked MHC variant, like DR4, from presenting that one crucial self-peptide that triggers rheumatoid arthritis without shutting down the rest of your immune system.
Targeted tolerance induction.
Or, conversely, imagine enhancing the ability of MHC class I molecules on tumor cells to present unique tumor-specific peptides, making those cancers glaringly obvious targets for your CD8 plus killer cells.
Boosting anti-cancer immunity.
Precisely. The challenges are immense, of course. You're tinkering with the absolute core of self-recognition. But understanding MHC in this detail is the first step, the potential to finely tune immunity by manipulating peptide presentation. It's one of the most exciting frontiers in personalized medicine.
A complex system with profound implications, from basic immunity to future therapies. Thanks for breaking that down.
My pleasure. It's a fascinating topic.